Okay, hello everyone. My name is Roman Simon. I'm a scientist at Boehringer Ingelheim Drug Discovery Sciences, and today I would like to take the opportunity to present you our approach towards MALDI-TOF Affinity-Selection mass spectrometry, which we implemented at our screening facility site to enable unbiased screening for protein-ligand interactions. And I start with explaining you a bit about what we are doing. So I'm actually part of the high-throughput biology group at Boehringer Ingelheim's research site in Biberach, Germany. And there we are responsible for the high-throughput screening of our in-house compound library against biological targets. For that, we are always interested in fast assay technologies, and our ambitions here are especially, of course, data quality, cost effectiveness, physiological relevance, and as mentioned already, speed. We have started a dedicated effort a couple of years ago to use mass spectrometry as a label-free readout technology for a number of reasons. And here our work and our main screening platform is the MALDI-TOF MS platform, which comprises the automated target plate preparation by the SciBiodisc 1536 multi-channel pipetta, and this is hyphenated with the Bruker RapiFlex MALDI-TOF device. We have used this platform quite successfully, and so far we addressed a whole lot of different enzymes coming basically from all major families of enzymes, with the only prerequisite that the in vitro reaction is compliant with the general rule that the molecular weight of the substrate has to be altered in course of the enzymatic reaction. And by that, we have targeted not only a lot of different and very diverse biological targets, but also different analytes covering a larger analyte space. And we are so far very happy with the performance of this technology platform, which we established in our group, and we also published the automation, the validation, and also the first application here. The first full deck screen formed in my group was a project example where we were looking or searching for inhibitors of the CGAS enzyme. We published this in SLAS Discovery last year, and here we were monitoring the transformation of ATP and GTP via CGAS to the cyclic product CGAMP. And as mentioned, this was the first full deck screen that was conducted using MALDI-TOF, and we used this platform to very efficiently perform this screen with excellent assay statistics, and we performed 1.1 million compound screen here with more than 7,000 dose response experiments in about 5 weeks for a project example where at that point no other assay technology was basically available. So as mentioned, we have already covered the in vitro enzyme field quite successfully, and we recently turned our attention then towards the next step in the drug development chain, let's say, or the lead discovery chain, which was then target engagement. And the question came up, can we use MALDI-TOF also to not only monitor the enzymatic conversion of the substrate to a defined product, but also to monitor in vitro target engagement, or more specifically, the binding of a small molecule ligand to its respective protein target. Okay, so we developed a concept based on Novartis speed screen platform, which comprises separation in by size exclusion chromatography in 384 well format. Here we have the protein target, which is combined with small molecule ligands or test ligands in individual incubations, and we also included a small tracer as a quality control for the sec integrity. The individual incubation mixtures are then passed through the sec material via centrifugation, which leaves us with 3 possible scenarios in these eluted fractions. In case 1, we have actual binding of the compound to the protein target. In this case, the whole protein-ligand complex is passed through the SEC material, and we are able to identify the ligand in the eluted fraction. In case 2, there is no affinity of the test compound to the protein target, which leads to the retention of the small molecule together with our tracer in the SEC material. And in case 3, we potentially can have a compromised separation, for example, by air bubbles. In this case, not only the test compound would pass the SEC material, but also our tracer. So this shows how we can use this tracer to detect falsified separation on the plate. As you can imagine, this is only a qualitative measure of protein-ligand interaction. And to give this also a quantitative aspect, we set up the second workflow, the hit confirmation workflow. Here is depicted 3 individual experiments for 1 ligand. We have a sample, which is the same as before. We have our biological target and a test compound and our tracer. And now we combine this with a low control experiment. Here we use an unrelated protein, for example, an unwanted protein isotype, or a completely unrelated protein like, for example, bovine gamma globulin. And we also include a high control experiment. Here we do a DMSO incubation, pass it through the SEC, and then we spike in a compound concentration after elution from the plate that mimics full 1-to-1 protein binding. By recording the mass spectra for all these 3 experiments, we can then relatively quantify the amount of bound compound with respect to its corresponding low and high control, and we call this then relative binding affinity. Okay, we came up with a couple of questions and challenges during this procedure. We first focused on the separation procedure itself and its characteristics, as well as small molecule detection and matrix interferences. So in the first step, we optimized the bed volume for the size exclusion chromatography resin in the 384 well plates. And we found that with approximately 70 microliter bed volume, we achieve stable retention of higher than 99.5%, which was then completely sufficient to proceed. We also, in this example, we used size exclusion chromatography resin with a molecular cutoff of approximately 10 kilodalton, and we used proteins of different size to show that if you approach this molecular cutoff barrier, the amount of protein that is passed through the SECs declines rapidly. So you should stay above this threshold. However, if wanted or necessary, you can of course use other size exclusion resins. This is very uncomplicated to exchange. In this combination, we looked at the stability of a plate, retention stability, and we found by using the QC peptide, so this quality control tracer, or fluorescein as a more simple tracer, we found stable retention of more than 99% across plates. We then looked for different matrices to maximize the detection rate of small molecule analytes, and we found by testing a handful of compounds along with the QC standard that HCCA gave the best signal to noise values, as well as the best and highest reproducibility. And in panel B, you see depicted the general or an example spectra in positive and negative mode ionization of a 5 compound mixture, together with the internal standard and the quality control reference for precise, which is the internal standard for the quality control peptide. As you can see, we still have a lot of background signals present. So the main focus here was to achieve as much mass precision as possible. And we do that by applying an external calibration prior to every screening campaign. And also we apply a quadratic internal calibration protocol using 3 reproducible background signals together with the internal standard and the QC ref signal to reach a mass accuracy of about 10 ppm across the campaign. With that, we tested 2 sets of roughly 1,000 and 5,000 compounds with diverse chemistry to assess the detection rate. And at a compound concentration of 10 micromolar, we have compound detection rates of about 85%. So this means we still lose about 15% of compounds. Nevertheless, in our opinion, it was worth to continue here as the possible advantage of having a high throughput label-free in vitro target engagement assay would actually compensate for that. So next, we focus on performance and reliability as well as functional relevance. Therefore, we had 2 reference compounds at hand when we choose the protein tyrosine phosphatate 1B as a validation project. Ref compound 1 is an active site ligand with a good binding affinity, which binds to the catalytic center of PTP1B. The second compound is an allosteric binder, which binds with medium affinity to this WPD loop region, and it stabilizes the protein in an inactive conformation. We confirmed the affinity and also the binding mode via ITC experiments and 2-dimensional NMR, which gave us also the opportunity to characterize potential hits in that came out from the screening campaign. So in the first step, we tested a compound of roughly 1,000, a set of roughly 1,000 compounds in 3 different and orthogonal binding assays, as well as in a MALDI-TOF-based PTP1B activity assay, and compared the results. Here we used, of course, the MALDI-TOF AS-MS concept together with an alpha screen competition format assay targeting the active site of the enzyme, and we also included differential scanning fluorimetry, where we looked for protein stabilization indicated by a shift in the melting temperature. When we looked at the individual hit sets, we found that the 2 reference compounds were only captured by MALDI-TOF AS-MS, along with only 1 additional hit. And we also found large individual hit sets for the 2 orthogonal methods, which had no overlap with the respective other methods. So this showed us that the MALDI-TOF AS-MS concept is a rather, let's say, stringent filtering method, but it seems to be quite reliable. We then compared our results with activity data, and here you see for the 3 MALDI-TOF AS-MS hits that we also were able to assess functional inhibition, and that this functional inhibition actually correlated quite well with the determined relative binding affinities. Notably, both other methods failed in identifying all 3 of these hits. To show that this also works for other protein targets, we also assessed the RBA values for reference ligands, for example, for CGAS, for carbonic anhydrase 2, human serum albumin, or the PTP1B isotype TC-PTP1B. And this in general works really nicely. This also demonstrates the versatility of the approach. You can basically adjust the assay just by exchanging the protein of interest. So next step was automation and of course the application in a fully automated run. And here we use a data handling process, which is based on the NIME analytics platform. So we use compound data, which comes from our in-house compound management. This is fed into a NIME workflow, which basically does reformatting and calculating of target masses from this initial CSV file into the MALDI Pulse 2.2 software compliant input files. Those are then imported into the MPP 2.2 software, and we use the synthesis screening workflow to perform MALDI measurements to generate result files for positive and negative ionization mode. These are then again fed into a NIME-based workflow, which does the data deconvolution and also applies the thresholds that we can predefine dependent on the campaign that we run. In general, we combine, because I said we work in 384 well format, we combine the 3 experiments plus a buffer blank into one 1536 well plate. This is then transferred to the automated MALDI spotting device, where the MALDI plates are prepared and then measured by MALDI-TOF MS. So this is how the NIME workflow looks like, and here in the end you get the result sheet, and here you have basically all the information that you need. Sample code, you also get information on the ionization mode, plate code, the analyte that was detected. We usually go for the M+H and also the first, second and third isotope, as well as the sodium adducts. We apply, or we have different qualitative parameters that are reported. For example, is the internal standard detected in all samples? Is the compound detectable against the buffer control in the first place? And also we have a qualitative parameter saying if there is actually specific compound binding or not. And then we have the initial value coming from the sample, compound binding in percent, unspecific binding from the low control, and this is then calculated into the RBA values. So automation was actually a big topic. This was one of the most challenging projects that we ever put onto our system. We had the ambition that we wanted to use our in-house high-throughput screening infrastructure. So usually in these formats, it's a very common approach to use compound pools, but we specifically wanted to go for single incubations on our regular HTS environment. So we did not have to adjust or integrate any new systems into our existing platforms. Of course, we had to schedule the automatic run. This was a bit difficult also because you have a lot of plate stacking, for example, but it's manageable, and we put a special focus on the transfer time of the filter plates after the samples were transferred to the filter plate, so that the actual incubations had minimum contact with the SEC resin before the complex is separated by centrifugation. If you look to the right, we have a Gantt chart, and here you see that we reach in this format plate cycle time of around 74 minutes for 1536 well plate, and this then translates into a theoretical throughput of 25,000 compounds per day. And this is actually also what we did. So we performed a full full screening day. We achieved a throughput of more than 23,000 compounds. We used the reference compound 1 as a constant high control in constant positive control throughout the campaign. And this was also constantly and robustly detected as an assay hit. We then performed 2 cycles of hit confirmation, which filtered out most of the initial hits and left us with a final hit rate of 0.021%, which were 5 confirmed hits out of 23,444 compounds. We also showed that it's potentially possible to record dose response experiments here. However, because we have to use relatively high protein concentrations, the sensitivity of the assay is usually not sufficient to discriminate higher compounds with higher potency. And we also found that the calculated RBA values correlate much better with inhibitory activity, or also with IC50 values, if you want so, than the dose response data from AS-MS with corresponding dose response data from, for example, activity assays. So for that reason, we usually compare RBA values to inhibitory activity if you want to do, or if we want to look for correlation of binding data with inhibitory data. So what we have demonstrated is that we have an automatable and HTS-capable high-throughput assay based on label-free MALDI-TOF mass spectrometry for in vitro target engagement. We have an analytical coverage of about 85% compound detection rate. There are different approaches how you can deal with that. So one would be that you use dedicated libraries with proven MALDI-TOF detectability. However, if you want to stay as chemically diverse as possible, for now at least, we accept the loss of about 15% compounds. We showed that we have a reliable detection of orthosteric and allosteric binders, which was not the case for more frequently used orthogonal methods. And the basically beauty of this whole technique is that we have a completely unbiased screening for protein ligand interaction, where we do not need any tags on the protein, we don't need any tracers, and in principle we are also independent of the knowledge on protein function. So if we have, for example, orphan targets, we can screen them as well as more defined targets, or targets for which structural information is available. So with that, we had basically checked all the questions and challenges that we came up with, and for now we can say that we have a label-free readout for small molecule target engagement, which we have already applied in several projects in-house. In total, we have been very open about the development and also the establishment and further development of our in-house MALDI-TOF-based platform. As mentioned, we have published the automation, the validation, and also the application of that. If you want to know more about it, please feel free to approach me anytime or check out our publications in SLAS Discovery. For now, we have successfully performed 3 full-deck screens in 2019 alone using this platform. We have expanded by installing a second device in the last year. And as mentioned, we also have successfully applied the AS-MS concept to several in-house projects, and we are working with this mainly in the with a focus on hit confirmation. But we are constantly working on improving this concept, as we think it's a very promising approach for early drug discovery. With that, I'm finished, and I would like to especially thank, of course, my whole team at Boehringer Ingelheim, from DDS, also from MedChem, the collaborators at Bruker, especially Maika Hamister, of course, as well as the colleagues from Analytic Jena. And if you have questions, I'm also happy to refer you to the Bruker exhibition booth if you want to talk with someone from the team. And of course, I'm also happy to answer questions if you contact me offline. Thank you very much.